I'm Ruth Slack, and I'd like to thank you all for being with us tonight, the final evening of Brain Health Awareness Week, which is hosted by the University of Ottawa Brain and Mind Research Institute. And if you missed any of our events this week, you can still watch these by going to the Brain and Mind website, where all of the links for each evening's events will be posted. So tonight we have a very special evening dedicated to our trainees who come from across Canada and around the world to work with our investigators here at the University of Ottawa Brain and Mind Research Institute. And because of your support, every year, the Brain and Mind Institute is honored to provide research scholarships and fellowships to its many trainees working in areas of our research priorities. And these priorities include stroke research, Parkinson's disease, mental health, memory and cognition, concussion, multiple sclerosis, neural dynamics, and also neuromuscular diseases. So funding for these awards are provided by generous donations and contributions by our community members who are committed to making a difference in brain health research, and also to inspire our future generations of clinicians and scientists to pursue medical research. So we would now like to take the opportunity to acknowledge and thank our many donors whose generous support has made such a huge impact in the lives of these trainees. These awards help to attract, again, the top trainees from across the country, giving them the opportunity to develop the knowledge and the skill to perform state-of-the-art research. This also helps to cultivate an enriched learning environment. And of course now, we would like to congratulate all our awardees from the past year. So let's watch the video that highlights our awardees.
Hi, my name is Michaela, and in 2018, I was honored to receive the Larry Hafner Fellowship from the PRC. I would like to send my deepest thanks to the Hafner family and to the PRC for awarding me with this. This award has allowed me to pursue my passion in Parkinson's disease research for over 3 years now. My favorite part of this award was how it allowed me to become part of the Parkinson's community outside of the scientific community. I loved being a part of PRC committee meetings and giving my input as a student, as well as being part of all the volunteer events and getting to interact with the members of the Parkinson's disease community. I'm happy to continue this research, and thank you so much.
Hi, my name is Karim Ibrahim. I'm a PhD candidate at Stephen Ferguson Lab, and I would like to take this opportunity to thank Mr. and Mrs. Marco Glesi for funding my graduate scholarships this year. The generous fellowship will help us advance our current research on Huntington's disease and other related neurodegenerative disorders. Thank
Hi,
you.
my name is Kim, and I'm very honored to be the recipient of the Saroj and Kishori Lal Family Scholarship. I was very happy and appreciative to learn that I was the recipient of your scholarship. Your generosity is allowing me to achieve my goals and dreams about doing research related to dementia and Alzheimer's disease. Thank you for your generous financial support towards my higher education.
Hi, my name is Michael Lin, and I just want to personally thank Mark and Gail Marcogliese for their fellowship and financial support. It's really allowed me to focus on new and exciting research into the mechanisms of mood regulation, the circuit mechanisms, and has eased the financial burden of pursuing a higher degree. So again, thank you.
Graduate students and postdoctoral fellows are the beating hearts of academic research. They are the ones who do the hard work, learn difficult and deep concepts, solve challenges, only to relentlessly push our knowledge boundaries. Supporting these trainees directly supports discoveries and new therapies, now and for the future.
So I'd like to thank the entire trainee committee members who helped to organize tonight's event. And you will also meet them as co-hosts in the Q&A panel. But for now, I'd like to pass the screen over to Leah Kayo-Bissonnette, who will host the next segment of our session. Thanks.
Thank you, Dr. Slack. Hi, everyone. My name is Leah Kayo-Bissonnette, and I will be one of your co-hosts for tonight's program. I am a third-year PhD student in neuroscience in Dr. Jean-Claude Bake and Dr. Len Mailer's lab, and my research focuses in cellular model of learning and memory. I'm excited to introduce 3 fellowship awardees who have enhanced their research abilities and learning experiences through funded research via donor contribution. Each speakers will have 10 minutes to present their findings and how the award helps support their research. For the first awardee, I would like to introduce Michaela Lunn, who was awarded the Larry Hafner Fellowship. She is a second-year PhD student in Dr. Michael Masher's lab, and her presentation title is The Role of Parkinson's Disease-Linked LRRK2 Kinase in Innate Immunity in the Context of Brain Health.
Hello. Thank you for having me here today. So my name is Michaela. As mentioned, I'm a second-year PhD student in Dr. Schlossmacher's lab, and in 2018, I received the Larry Hafner Fellowship from the Parkinson's Research Consortium, which, as you saw, I'm very thankful for. So today is taking a look at some of the work that I've done looking at the role of LRRK2 in innate immunity as it relates to brain health. So in our lab, we study Parkinson's disease, which I'm sure most of you are familiar with. So it presents sort of like this motor disorder. You see the gait, tremors, but it also is associated with many non-motor symptoms, such as depression, constipation, and other symptoms like this. So in our lab, we believe that Parkinson's disease is a complex disease. And what I mean by that is it requires some sort of genetic susceptibility, so a gene mutation, along with some sort of environmental trigger. So an infection, possibly toxins, different experiences that you might encounter that leads to the onset of Parkinson's disease. So there are many genes that have been associated with Parkinson's. And as mentioned today, I'll be looking specifically at LRRK2. LRRK2 is one of the most commonly associated proteins with Parkinson's disease. And until now, much of the research has looked into its role in the brain. However, in our lab, we believe that this protein might actually have a role primarily in the body. And we think that Parkinson's disease might actually onset in either the nose or the gut. These are 2 places that are at the interface of the environment. So it's where our body first encounters the things in the environment that we might breathe in. So for example, infections. And leading from that, we actually believe that LRRK2 may have a primary role in the innate immune system. So the body's first defense in fighting infections. So this protein, this is a schematic of the LRRK2 protein. It's very large, and as you can see, there are multiple mutations in the protein that can increase your risk for developing Parkinson's disease. I've circled one here, G2019S, which is the most common mutation in humans that can lead to Parkinson's disease. And this increases the kinase activity of the protein. And very simply, what a kinase does, it allows one protein to basically activate another protein, and then this will activate a pathway. So basically a kinase function allows LRRK2 to activate different pathways. So with an increased kinase activity, this mutation allows LRRK2 to be overactive and to have an overactive kinase function. So we have mice that have a G2019S mutation that we use to study this in the lab. Similarly, we have mice that have a D1994S mutation, and what this does is the mice have no kinase activity at all. And I'll just point out quickly that this does not occur in humans. It's not associated with Parkinson's disease. It just allows us to study the role of the kinase activity specifically in this protein. And why that's very important is that in human trials, there has been developed LRRK2 kinase inhibitors as a treatment for people with Parkinson's. And because we believe this protein is important in the immune system, we want to make sure that inhibiting the kinase activity is in fact safe. Next, I'll point out that there's actually been mutations in LRRK2 linked to Crohn's disease and leprosy. two diseases that are seemingly very different from Parkinson's disease. And both these diseases are characterized by inflammation, and they both require some sort of infectious hit to onset disease. So this kind of supported our theory that LERK2 linked Parkinson's might also require some sort of environmental or microbial hit to onset, and that it would be characterized by inflammation. So the objective of my study, and this is what was outlined in my application when I got the fellowship from PRC, was to identify the physiological role of LERK2 in the immune system. And specifically, we hypothesized that one, this protein is involved in the innate immune response, and that variants or mutations in LERK2 can modulate or change the outcome of immune response. And secondly, this kinase activity of the protein is required for its role in the immune system, and that if we inhibited that kinase activity, we would lose any immune effect that we would see with normal LERK2. So first, we employed a sort of infection model that allowed us to look at brain health. And what I mean by that is that we infected mice that were only 1 day old with a virus called reovirus. And this virus in a young pup model actually causes encephalitis in the brain, so brain inflammation that can be fatal. So we inoculate the pups by putting the virus on the nose, they breathe in the virus, and it eventually reaches the brain where it causes encephalitis. And our two main readouts were first PFU, which is plaque forming units. And what that is just a count of the amount of virus that's in that given organ. So measuring the amount of virus, the viral burden is what I'll refer to it as. So we're looking in the lung and the brain at 2 different time points, 3 days after infection, so early, and 11 days when it's later in the disease progression. And lastly, we measure survival of the mice. So first we looked at LERK2 knockout, so mice that have no LERK2 protein at all. And the higher the points are on this bar, the more viral burden they have, the more virus in that organ. And first, when we looked at viral burden, we didn't see a difference between normal wild type mice or the mice with no LERK2. However, when we looked at survival, as we expected, especially seen in the females, but seen in both sexes as well, mice with no LERK2 had higher death. So they were more susceptible to the disease. So this told us that the LERK2 protein was important in the course of reovirus infection, because when mice didn't have that protein, they died more likely. So we next wanted to look at this PD-linked mutation, the G2019S, which is overactive. So as we expected or hypothesized, we did find that mice with this GS mutation had less viral burden than normal mice. So this overactivity allowed them to control the virus better. And this occurred in the lung and the brain. However, interestingly, when we looked at the survival in these mice, and specifically in the female mice, we actually found that female mice that hold this G2019S mutation actually had more death. So despite the fact that they controlled the virus better, it actually led to more fatality in these females. So what we hypothesize is happening with this mutation is that they're overshooting their immune response. So although they're able to better clear the virus, the inflammation is too sustained and not controlled, and it actually is harmful in these females. So next we looked at the kinase activity of these mice. So looking at the D1994S mice that have the LERK2 protein, but it does not have the kinase activity. And as we suspected, based on the previous results, when there's no kinase activity, they did have heightened viral burden. So in the lung, the mice with no kinase activity had more virus than the normal mice. We didn't see differences in the brain, however. Again, in an interesting paradox, when we looked at survival, this increase in virus didn't translate to a survival deficit. In fact, these mice here are the ones without kinase activity, and they actually were protected and had heightened survival compared to their wild type counterparts. And again, this is especially seen in the females. So if you're seeing this consistent trend that the females seem to be more affected by changes in this protein. So this data was really important because it actually supports the use of these LERK2 kinase inhibitors in humans, because this data suggests that not only would inhibiting the kinase not be harmful, but it actually could be protective in the context of the immune system and against infections. We're still repeating this, but it's exciting results nonetheless. So next we wanted to look at where is the role of this protein in the periphery versus the brain. So we really wanted to see, as I mentioned, much of the research presently is looking at its role in the brain, but we really think it could be in the body predominantly. So what we did in this paradigm is we used the exact same virus, reovirus, causing encephalitis, again in a young mouse pup model, except this time we administer the virus directly to the brain with an intracranial injection. So in the first paradigm, the mice breathe in the virus, it's the body first and then the brain. So we don't really know where LERC2 is playing the predominant role. By putting the virus directly in the brain, we hope to minimize the amount of peripheral involvement. So we can't completely count it out, but we're hoping to limit it to just the brain. And then we also know exactly how much virus got to the brain. We measured viral burden in this paradigm as well, but for the sake of this presentation, I'll only be showing survival. So interestingly, when we looked at the G2019S mice, and I'll point out quickly that in this paradigm, there's 100% lethality. So it's measured as time to death. So you'll see there's no survivors in any genotype just because of the way that this paradigm is. But more importantly, you'll see that there's no genotypic differences at all in these mice, the wild type versus the G2019S mice. And why this is interesting is if you recall, when the exact same virus was administered to the nose systemically, we did see a survival difference in the females. But now when it's given directly to the brain, we don't see any of these changes. So this would suggest to us that in the context of reovirus, the LERC2 G2019S and even the wild type LERC2 is working predominantly in the periphery or the body rather than the brain immune system. Similarly, when we looked at the kinase dead D1994S mice, we see no survival differences at all again. And if you recall, in this paradigm, we actually saw stark survival benefit with the DS mice in all sexes and when they were pooled, and now we don't see any differences at all. So again, this suggests to us in the context of this reovirus encephalitis model, the LERC2 protein seems to be working predominantly in the periphery to fight the infection rather than in the brain. So the next steps of this project, and in the context of time, I wasn't able to show our preliminary results, but I'm happy to talk about it at another time. We are repeating this in an adult mouse model. So of course, we want our work to be as translatable as possible to human PD patients. And of course, our other work uses P1 mouse pups, which isn't exactly translatable. So we're recreating this in adult mouse models. First with vesicular stomatitis virus, which causes encephalitis, the exact same as reovirus. And then secondly, we've started using influenza A virus, which causes pneumonia. And we actually did this with COVID-19 in mind because LERC2 is most highly expressed in the lungs. And we actually wanted to see if this protein could be protective in the context of a disease that's mainly in the lungs. So that's some interesting results that we've started to look at recently. So for my overall conclusions, I'll leave you with this. One, that the wild type LERC2 protein functions in the immune response independent of its kinase activity. So our original hypothesis that the kinase activity was critical was actually wrong, but that was hopeful because it actually suggests that using these LERC2 kinase inhibitors in humans will be beneficial and safe. Secondly, that LERC2 behaves with a female sex bias in the immune system. This is especially interesting because Parkinson's disease in general affects males mostly, but LERC2 related Parkinson's disease is actually more common in females. So this female sex bias is actually consistent with human data. And thirdly, that LERC2 functions in the immune system predominantly in the periphery. And this is interesting results, and we really hope that we can help to shift the field's view of LERC2 and maybe even Parkinson's disease onset outside of the brain and into the body. So I'll leave you with this final model where we suggest that the G2019S mutation, these mice have too high of an inflammatory response. So the inflammation is too much, it's uncontrolled, and it's harmful to the host. Whereas in knockouts, they have too little inflammation without the LERC2 protein, and then they die from the virus because they can't fight the virus. Where wild types fall somewhere in between, where they have enough inflammation to fight the virus, but not too much that it's harmful to the host. So that's it for my presentation. I want to give a heartfelt thank you to the Parkinson's Research Consortium for funding me for the past 2 years to do this research. And a big thank you to my whole lab for helping me with this work. Thank you.
Thank you, Michaela, for your presentation. That was great, and congratulations for your award. So our next awardee has won the Mark and Gail Marcogliese Graduate Scholarship for Research. He is a 6-year PhD student in neuroscience. He is supervised by Dr. Jean-Claude Beek and co-supervised by Dr. Len Mailer. His name is Michael Lin, and his presentation title is emergent neural computation from novel recurrent circuit dynamics in a mood-related network.
Hello, yeah, thank you so much, Leah, for the introduction. So I'll just share my screen. All right, yeah, so thank you so much. I'm really happy to be here and to talk about my project. It should be a pretty exciting set of results, hopefully. So as a lab, we're really interested in the dorsal-raphe nucleus. And so this is a nucleus that secretes serotonin throughout the brain, which is involved in regulating mood. Obviously, antidepressants target serotonin receptors. And it's a really interesting nucleus because it's a hub nucleus. So it receives lots of inputs from all over the brain and also sends its serotonin output to the rest of the brain. And so really we're trying to figure out fundamental properties as to how information transmission through this hub works and how that can be related to behavior. So these are just some of the areas that dorsal-raphe nucleus talks with that are shown to be involved in emotion and affective processing. So another area we've become involved in is looking at lateral habenula. And so this is a small area right below hippocampus, and it effectively represents acute threats and is implicated in depressive-like symptoms. So for example, this is a recording in monkeys showing single unit activity, so single neuron responses, showing that if you deliver an air puff to the monkeys, these lateral habenula neurons start firing like crazy. So it's a really acute representation of threats. Also, for example, looming eagles would set off lateral habenula. That's been shown, foot shocks, this kind of thing. And it's also been implicated in depressive-like symptoms. So here, these show bursting in C of these neurons, so really high firing rates in response to depressive circumstances. And so really the question that my talk is focused on today is, how does graded threat level, represented by habenula input, modulate mood and this serotonin output to the rest of the brain? And so a key methodology that I'll be talking about today is single cell electrophysiology. And so if you look here on the left, essentially what we do is we inject an opsin, so a light-gated ion channel that allows us to isolate one input, in this case, lateral habenula. And so we inject this, we wait several weeks, and then we make midbrain slices, shown here in the middle, of dorsal-raphe nucleus. And then we attach onto genetically identified serotonin neurons with a small glass electrode and record voltage or current that's passing through these neurons to try to get a sense of how the circuit actually works. And later on, we're going to translate this into behavior. That'll be the last part of my talk, but this is sort of the core method that I'll be talking about for a while. And I should also mention, we can use light then to activate particular inputs while recording these. So we can record a serotonin neuron and particularly activate an input from habenulins and look at processing of this. So when we do this is the same setup I've told you about. When we stimulate and activate habenula inputs at low frequencies, so single pulses, we see something really classic that's found in the brain, which is this nice inward current. So here we're recording in voltage clamp. So we're clamping the voltage of the neuron at minus 70 millivolts, and we're looking at the current flowing in and out of the cell. So this is an inward current, so it's excitatory. And we show can be blocked with the drug NBQX, meaning it's AMPA receptor mediated transmission. So this is really classic excitation. We also show it's monosynaptic by putting TTX, which is another drug blocking sodium channels, and then recovering with 4-AP. So far this looks like really just classic excitatory neurotransmission. So habenula excites raphe. However, we saw something really unexpected, which is if you excite these habenula terminals at really high frequencies, you actually trigger the opposite, you trigger inhibition. So again, same setup. Single pulses, I already showed you, excitation, this nice inward current. Here we're delivering a 20 hertz pulse train and we show an outward current. So very odd. And so effectively what we show here is by putting a selective blocker, WEI-100-635, is that this outward current or inhibitory current is mediated by 5-HT1A receptors that couple to GERCs to mediate this inhibition. And so this is really interesting because 5-HT1A receptors are classically thought to be autoreceptors. So they're thought to mediate a neuron's own self-regulation of its excitability. So if a neuron fires too much, it secretes serotonin in the extracellular space. This is what's thought, and it inhibits its own firing rate. So that's one model, but another idea is that maybe these serotonin neurons are organized in a recurrent network. So this is shown below here. And if this idea were true, then these serotonin neurons would inhibit one another effectively. Some converging lines of evidence have indicated that this is actually the case. I'm just going to show you probably the strongest evidence, but there's convergent anatomical and physiological evidence for this. So here we're injecting another opsin, but directly into dorsal raphe nucleus. And we're injecting it at really low doses. So effectively, if you look, the green neurons will express this opsin, and you get a mix of green neurons expressing the opsin and red neurons that don't express the opsin. So you get a sort of a salt and pepper mix. And so you can ask what happens if you record from a green cell and stimulate it with light. And you see over here really nice spiking responses. So these neurons can be stimulated, the green neurons. But what if you record from a neuron that doesn't actually express the opsin? In that case, if you stimulate, if you look on the right here, you see the exact same outward current that we showed previously. And we show it's the same, it has the same kinetics, it can be blocked with the same drugs, etc. And so this really conclusively shows that the raphe is organized in this recurrent network. So serotonin neurons actually inhibit one another instead of inhibiting just themselves. So the next section is very exciting. So here we try to look at rules for information transfer through this circuit, which turns out to be really cool. So it turns out that there's really striking short-term dynamics of these recurrent connections. So remember, the entire network is getting excitatory input, and then these neurons actually talk to one another and inhibit based on some rules that we're going to talk about. So here, slightly different paradigm. So we're still looking at raphe, and we're still looking at serotonin neurons, but here we're electrically stimulating other axons coming from other serotonin neurons. And we have a cocktail of drugs here to block all other responses effectively. And so we show when we do this, we can get an identical outward current that we've seen before. It's also blocked by Wei. It has the same IV curve, same kinetics, etc. And what we see is something really striking, which is here we're doing a frequency experiment. So here we're keeping the same pulse number, so it's always 8 pulses, and we're varying the frequency. And what we see here is if we stimulate at 1 hertz, we get absolutely no response at here. So you can see in this zoomed in panel below, there's just no response after each stimulation. However, as we increase the stimulation frequency, we get a really dramatic increase in the response until it's a really large response at 20 hertz. So this is just some group data showing that you get some boosting at really high frequencies. So this is really odd. So what we did next is effectively try to mathematically explain the results that we've obtained. So unfortunately, we can't spend too much time on this, but we ended up describing the results I just showed you and some other results with a unified model of short-term plasticity dynamics, which is called a linear nonlinear model that unifies results across multiple timescales. So we can explain this really odd nonlinearity that we saw, where short or low input frequencies don't trigger the recurrent inhibition, and high frequencies trigger it really strongly. So this is just to show you some data. So on the left, we see the charge passed, and I just want you to focus on the experiment in black and the model in blue, and there's a really good fit. And on the right, we tried to use the same model to explain a completely different set of experiments that weren't fit at all, and it explains it almost perfectly. So it's a really good fit for these odd dynamics. Next, we focused on some network modeling just to see, again, the rules of information transmission through this circuit. And unfortunately, again, we can't spend too much time on this, but just to give you a taste, here's some simulation showing that short-term plasticity, so the model that we fit, actually generates really interesting input gating rules. So what does that mean? Well, turns out if you have two inputs to the network, so say one of them is habenula, another is, let's call it input A, habenula can actually gate access to the network from the other input. So it can inhibit the other input from actually triggering network responses. So we're gonna look at this. On the left here is just when input A is active, and on the right is when input A is active and it's preceded by a really strong habenula input. And so you see here, so this is just a slide with the STP dynamics, the short-term plasticity dynamics intact. And you see here on the left, A now triggers a really nice response in the serotonin network, shown on the left here. And on the right, if it's preceded by a habenula input, you actually get almost complete suppression of the response that A is triggering. And so this is shown on the right by this input-output transformation. So effectively, without habenula input, you have a really nice input-output curve shown in black. And with a habenula prepulse, you get a really dramatic lowering of this input-output curve. And we also show that's almost completely dependent on short-term plasticity dynamics. So if you keep the same recurrent network, but just take out the dynamics that I just showed you, this effect goes away. So effectively, STP generates these really interesting input gating rules. So far, the conclusion is, at low input frequencies, there's direct excitation that dominates. And at high input frequencies, there's this recurrent inhibition that dominates through these really odd short-term plasticity rules at these recurrent synapses. So the relevance for mood might be graded threat processing. So just to show you a little bit of behavior, we wanted to translate these findings to an actual behavioral model. So we developed a head-fixed classical conditioning task. Here, what we're actually doing is injecting an opsin in habenula and putting an optic fiber in raphe. So that we can stimulate this pathway in an actual behaving animal and look at what happens. So it's a really simple classical conditioning task. The animal hears a tone and gets some delayed reward for that tone. And it will learn to lick after the tone because it anticipates the reward. And we're going to look at whether we can modulate this anticipation by activating the habenulo-raphe pathway at various frequencies that we've mapped onto various network responses based on our modeling, effectively. And synaptic physiology. So this is what we're going to do. So I'll just show you two quick videos. The first one is just the baseline response. So if you look at the cheek, you can see some licking. So you'll see tone. So the cheek, if you look, the animal's licking, he's very excited. Then he obtains the reward. Then he just goes crazy and he's extremely excited. Next, we're going to try to activate the habenulo-raphe pathway at really high frequencies. So watch what happens here. And again, watch the cheek of the animal. So the tone happens, the animal completely stops what it's doing. It has this pseudo freezing behavior, and then it continues reward consumption afterwards like nothing happened. So we also show this isn't dependent on the motor effects, and it's a really robust thing. So just to show you some summary data, stimulation during a trace, at high frequencies, 20 hertz, there's a really significant depression of this anticipation of licking. But at low frequencies, you get no change. So we think that this actually maps really nicely onto the circuit dynamics that we talked about earlier. Just to show you what happens just after that stimulation is the animal consumes reward normally. So there's no long-term change effectively from this. So just to conclude, there's a recurrent inhibitory network of serotonin neurons in raphe that operates over recurrent timescales, over behavioral timescales, and is governed by these really interesting short-term plasticity rules. And we've shown that this recurrent network can actually dynamically filter aversive information from habenula to modulate behavioral anticipation. And so for that, with that, I just want to thank everyone involved in this project, but especially my supervisors, Dr. Jean-Claude Bake and Dr. Len Mailer, and Dr. Richard was also involved in some of the modeling here. So yeah, thank you, everyone.
Thank you, Michael. Really cool result you presented today. Congrats, really great research.
Thank you.
So without further ado, I will present our third and last presenter for this segment. So it is Jared Lau, who has been awarded the STAR Award. He is a second-year master's student in cellular and molecular medicine. He is supervised by Dr. Hans Lockmuller, and his presentation title is Rare Neuromuscular Diseases / Finding Genetic Causes of Congenital Myasthenic Syndromes.
Hi everyone, thanks for that introduction. So let me get my slideshow started here. All right, so first off, thanks to all the organizers for putting on this event. It's a great chance for us to, get to tell everyone about our research. I'd also like to thank the Center for Neuromuscular Disease and the uOttawa Brain and Mind Research Institute for supporting me through their scholarship in translational research, which I got earlier this year. So thanks so much for that. So today I'll be talking to you about finding genetic causes of congenital myasthenic syndromes. So first, a bit of background on the important biological structure of congenital myasthenic syndromes, or CMS, which is the neuromuscular junction, which I'll just refer to as the NMJ because it's a mouthful. So this is basically the connection point that your brain uses to send a signal to the muscle to tell it to contract and move your body. And this transmitted signal has to have a consistent strength and duration to produce reliable muscle contraction. And this process is really highly regulated by a complex network of biochemical pathways that involves a ton of genes, as I've kind of depicted here in this diagram. So these biochemical processes are performed by proteins, which are encoded for in your DNA. Your DNA provides the instructions for your cells to make these proteins, and when mistakes or mutations happen in these instructions, it can lead to malfunctioning proteins. And when these mutations affect proteins at the neuromuscular junction, which regulate one of these processes that sends this signal, we can get something that looks like congenital myasthenic syndromes. So CMS are basically a group of rare and inherited neuromuscular diseases with a global prevalence of about 1 in 300,000. The patients are pretty recognizable by a few common symptoms, such as ptosis or drooping of the eyelids, as you can see in these patients here. They experienced exercise-induced muscle weakness, which is a really classic symptom of CMS. They often have a reduced muscle bulk because they're not using their muscles as much. And in severe cases, they can even have a really hard time breathing. Now, there are 33 known CMS genes, and even though we know that there's so many that have been linked to CMS, still about 40% of patients that are provided with a clinical diagnosis of CMS, they still go without a genetic diagnosis. And this is really critical to the determination of an appropriate therapy for these patients. Now, the proportion of patients that haven't received genetic diagnosis also points to the fact that there's more to know about this disease. And that's where my project lies, in the discovery of new genes in CMS. And this can help us develop better diagnostic tools and identify new targets for therapies. So how do we study rare neuromuscular diseases in CMS? So my project basically takes a translational research approach where we collect data from patients and use that information to research how CMS works, and then feed that information back to clinicians and patients to develop customized treatment strategies. So the aims of my project are to, 1, identify a candidate CMS gene, and then to 2, validate that CMS gene using cell and animal models to see if we can kind of produce results that look something like CMS. So how do I identify rare gene candidates in the first place? Well, I take genetic data from groups of CMS patients, which have had their data deposited into rare disease databases, such as the one we use, which is called RDConnect. And then I use clinical information and a set of data filters, which basically account for the normal genetic variation between CMS patients and healthy individuals. And then I go through each patient case by case and generate a list of top candidates, which I then research in cell and animal models. So using this strategy, I basically analyzed two sets of patients. The first cohort of patients, they all had a pretty broad range of CMS symptoms, like the classic ones I mentioned, like fatigable muscle weakness. But the second cohort all specifically had episodic apnea or troubles breathing. So after this analysis, we found that quite a few of them had mutations in genes that were already related to CMS, which can be seen in the kind of rainbow section of my pie charts here. But what I'm really interested in are these groups labeled VUS, or variants of unknown significance. And this is basically just a group of genes that have potentially damaging mutations, which have not yet been linked to CMS. So that's where I get my two top candidates for a new CMS gene, MGAT5B and CYT11. So in order to study the effects of these gene candidates, I use what we call morpholino oligonucleotides to essentially just block the production of proteins like MGAT5B and CYT11. So the morpholino oligonucleotide just basically binds to DNA and prevents the cell machinery from making these proteins. So I inject these morpholinos into zebrafish eggs, and then I observe the effects using a number of different collection methods. So we do these experiments in zebrafish because they're translucent during their development, and so we can see directly in and be minimally invasive. So the first tests that we use are behavioral assays. So we measure the activity of injected embryos while they're still in the chorion or the zebrafish egg. And this basically just acts as a window as to how well their neuromuscular systems are developing. Since the chorion is translucent, we can just record these with a camera, and then we extract data. So you can see the fish are just twitching and then kind of rotate around in the chorion there. So we record the rate at which these movements occur and the duration that these movements last. So our first knockdown experiments with MGAT5B, we can see that there's actually a slight increase here in the rate at which these movements happen. And although it's interesting, this actually kind of works against MGAT5B as a CMS gene. But when we repeated these experiments with CYT11A and B, which are the zebrafish gene equivalents of the human CYT11, we actually found two really interesting things. Is that the rate of these movements significantly decreased when we knocked down CYT11A and B simultaneously. And then the duration of these movements was also significantly decreased. So this is a great sign that these fish have big neuromuscular issues. So the next steps in my project is to measure these embryos' response to touch once they've hatched. So by this stage, the neuromuscular systems are much more developed, and we can measure other things like how fast they can accelerate, their top speed, and the distance that they can travel. And again, this is easily recorded by video. And you can see here, you just poke them on the back of the head, and we record how they move and analyze this data using software. But you could see that some of the fish had a pretty hard time swimming away. Rather, they kind of like swim in circles or just spasm. And this is a pretty classic response from a zebrafish with some sort of signal transduction abnormality. So I did actually complete these experiments just recently, but I'm still in the process of analyzing that data. So, I'll keep you all updated with the results from those experiments. One other thing I'd like to mention, one of the future experiments that we're going to be doing with these fish is to actually measure their muscle fatigability, which, as I said before, is a classic symptom of CMS. So one of the postdocs in our lab, Emily, which you'll hear from later, is actually designing an assay where we can measure the muscle fatigability of these fish of up to 96 fish at once. So that'll be really cool once we get that up and running. So the last thing we do is use fluorescent labeling to look at the structure and formation of the neuromuscular junctions. And from these pictures, you can see we take two differently colored labels specific to muscles in green here and nerves in red. The overlap produces this yellow color, which basically indicates the locations of all the neuromuscular junctions. And then we can extract data from these pictures, such as the number of neuromuscular junctions, the surface area of these of the contact points, the length of the motor neurons, how often they branch from each other, et cetera, et cetera. So in these experiments, things like a reduction in any of the things that I just mentioned could point to some sort of neuromuscular issue. So in conclusion, from my data this far, we can say that CYT11 is probably a stronger gene candidate for causing CMS. And based on the function of this gene in vesicle trafficking, as found by other studies, this could be occurring through a mechanism where we think that synaptic vesicles build up and release all their acetylcholine at once. And this is basically too much acetylcholine for the muscle fiber to handle and will make it less sensitive to sequential stimuli. And that would cause the fatigable muscle weakness that you see with CMS patients. So validating CYT11 as a CMS gene will really help clinicians diagnose future patients with CMS through a more thorough genetic screen and allow the prescription of appropriate therapies for patients with mutations in CYT11. So with that, I'd just like to thank all the members of the Lockmuir Lab, the CHIO Research Institute, and all the supporters of the Center for Neuromuscular Disease and the uOttawa Brain and Mind Research Institute. Thank you for supporting me through your STAR Award. So yeah, thanks.
Thanks, Jared. I've actually never really heard about this disease before, and it's really interesting to learn about it through your presentation and research project. Thank you. Okay, so thanks to all of our speakers for providing such informative talks. I'm now going to hand over the screen to my co-host, Kevin Smith.
Okay, thank you, Leah. Hello, everyone. My name is Kevin Smith, and I'm a fourth-year MA-PhD student currently in Dr. Nafisa Ismail's lab. My research focus is in the impact of stress on mental health during pubertal development. Tonight, I will be highlighting 10 trainees through a rapid-fire session. This series of presentations will provide the speakers with an opportunity to showcase their work, highlighting the research pillars of the Ottawa Brain and Mind Research Institute in just under 3 minutes. I hope that you enjoy. First, I would like to introduce Emily O'Connor. She's a postdoc researcher with AFM Fellowship in Dr. Hans Lockmuir's lab, and she's going to be talking about childhood disorders with disturbed neuromuscular transmission, from gene discovery to personalized treatment.
Okay, fantastic. Hopefully my screen is being shared now. If you can all see that. Is that, sorry, could someone just confirm if it's working? Because my screen is
confirmed, yeah.
Okay, perfect. Thank you so much. Okay, so hi everyone. I just want to say thank you for all taking the time to listen to our talks. So my presentation will continue on from Jared, who you just heard from, because we're both in the Lockmuir lab studying rare neuromuscular diseases. So he told you about gene discovery and about generating zebrafish models to determine whether different genes might cause a certain disease. And now I'm going to show you the sort of next steps in this process. So this is where we can use these models to test different treatments. So this is a really important step because 95% of rare diseases actually lack FDA approved treatments. So And obviously this is affecting a huge number of people, so over 400 million people globally. Okay, so one of the things I wanted to touch on before we analyze zebrafish, we can actually do a really cool technique in our lab where we take skin cells from patients and we can make nerves from them. So we can also make muscles as well, and patients seem to prefer when we take skin cells rather than nerves and muscles from them. And what we can do is we can take these cells and we can convert them in the lab into neurons in less than 1 month. So you can see here we have nerves growing across a plate. And this is really interesting because we can then test drugs and see how patients' individual cells respond to treatments. And then if we find drugs that show promising results in patient cells, then we can go back to our zebrafish models and try the drugs on those as well. So fish are actually fantastic models for testing drugs because they develop really quickly. You can see here in just 90 days, we have breeding adults. They're also transparent. So when they're young, we can actually see through the skin and look at their muscles and their brains. They're also very small and they perform different movements, as Jared touched on, so that we can measure their responses to different diseases and also to different drug treatments. So just as an example here, here you can see a 2-day-old fish. When we touch it, it responds to the touch. Here's a fish with a disease that can't respond appropriately. And then once we've treated the fish with a drug, we can see its response to movement there. We can also do this on a larger scale. So at the minute, we're optimizing this protocol where we can screen 96 zebrafish at once. We can analyze their movement in response to different drugs, and we can get this movement output here. You can see the red traces, and we can do things like track how they move over time. So this is just an overview of the workflow that we use all the way from gene discovery to treatment testing in rare neuromuscular diseases. And I'd like to thank you all for your attention.
Okay, thank you very much. That was very interesting. So the next speaker here is going to be Emerson Harkin, a PhD neuroscience student in year 2, is supervised by Dr. Richard Note, co-supervised by Dr. Leonard Maller. And the title is going to be Reverse Engineering the Serotonin System. It's important to note that the work presented was carried out in close collaboration with Dr. Jean-Claude Baig.
Great, so hi everyone. Hopefully you can see these slides, and I trust that someone will let me know if not. Yeah, so I'm Emerson. I'm a PhD student with Richard No, and I'm going to be talking about reverse engineering the serotonin system. So you've already heard about the serotonin system a little bit this evening, but just to recap, the serotonin system actually consists of a really small number of neurons, some of which make serotonin and some of which don't, located in the brainstem. And even though, together these two groups of neurons comprise only a fraction of a % of the cells in the brain, they play a really big role in regulating a wide range of animal behaviors. So, in particular, if you put yourself in the shoes of a mouse, you might be really worried about where your next piece of cheese is going to come from, or whether there's maybe a scary cat around the next corner. And serotonin seems to be pretty important in basically integrating information about the closeness of the next piece of cheese or the scariness of a nearby cat, putting that all together in the brainstem somehow, and then exporting that information to the rest of the brain to guide you towards running towards the cheese or running away from the cat. So fair enough, it sounds kind of simple, but it turns out that in reality, we don't really understand very well what is going on in the middle here. So to kind of shed light on this process, working together with Dr. Baig, I carried out some experiments to basically map out the electrical properties of the main two types of cells that are in the serotonin system, and then working together with Dr. No to plug those results into a series of computer simulations. We learned a few things about how this system might work to guide behavior in real animals. And the things that we learned can pretty much be boiled down to this one kind of sentence that it turns out the serotonin system is a little bit like a radio in a couple of ways. It receives this kind of messy information from all over the brain about cheese, cats, and all kinds of other things. It sends a clear signal to the rest of the brain about what to do. There are a couple of mechanisms in the cells that actually make serotonin that act to select the kinds of inputs that it's going to respond to tune into different types of inputs if you want. And lastly, we found that the interactions of a common type of cell in the serotonin system that actually doesn't make serotonin itself acts kind of like a volume knob on the output of the cells that actually do make serotonin. Now that last point we thought was kind of interesting because a lot of antidepressant drugs basically exploit kind of a similar mechanism to kind of increase the output of serotonin throughout the brain. So we think that maybe this mechanism could be interesting to make better antidepressant drugs at some point. But that's kind of the long range view of our work. So with that, I want to thank everybody who contributed to this project.
Hello, everyone. It looks like on my end here, the screen had froze. I guess we'll move on to the next speaker because it doesn't look like it's coming up here.
Apologies, we're trying to stop it now. There we are. Back to you, Kevin.
Thank you. So I'll definitely have to start thinking of the serotonin system like a radio from now on. I like that idea. So our next speaker here is Matthew McDonald. He's a postdoctoral fellowship in his 5th year, working with Dr. Grade Selassie. The title is called Visualizing Invisible Strokes, Studying Microinfarction in Mice.
All right, thank you very much for the introduction. So yeah, today I'm going to talk to you about microinfarction and how we're trying to model that in the lab by using rodents. So first of all, what are microinfarcts? Well, microinfarcts are small mini strokes. They're found in approximately 30% of healthy individuals over the age of 65. They're associated with a twofold increased risk of having a larger, more impactful stroke. And microinfarcts often occur in parallel with many other vascular diseases. So how are microinfarcts detected in humans? Well, first and foremost, they often just go completely undetected because most microinfarcts appear to be asymptomatic. However, with advancements in imaging techniques, and if you're lucky enough to actually have an MRI, you can visualize a microinfarct. So these are 2 MRI photos looking from the top down as well as the side. And you can see in the top left of both images, if you look really closely and in the right spot, you can actually visualize that small microocclusion, that small microinfarct. Because it's so difficult to detect microinfarcts, often they're not diagnosed until post-mortem autopsy. And even then, they're vastly underrepresented. So for example, if 9 microinfarcts were detected in a brain pathology sample, actually it's estimated that there could be up to almost 5,000 microinfarcts within the brain. Therefore, it's very difficult to study how microinfarcts impact brain health in humans for this reason. Therefore, in our lab, we've developed a mouse model of microinfarction where we surgically inject fluorescent microspheres into the brain circulation. they ultimately become lodged in small blood vessels and produce these small microocclusions. And you can visualize each of these green microspheres within the left hemisphere of this brain. And then in our experiments, we can actually dissect out that brain. We can mount it on a vibratome where we can section the brain. We've adapted this technique where we actually mounted a camera at the top and then also a blue light. So we can actually excite these fluorescent microspheres with the blue light. And then while we're sectioning the brain, we can actually take pictures from the top down. So we'll get a bunch of images as we section the brain that look kind of like this image on the top right, where you can see where each microsphere is located. Using these series of images, we've developed an automated technique where we can actually automatically detect where each microsphere is located and then produce a 3D rendering of where they're located within the mouse brain. So we can actually determine where each microsphere is located to any given level of the mouse brain atlas. So now that we've localized and actually visualized those invisible strokes, we can perform a bunch of different experiments. For example, those sections that we sectioned on the vibratome, we can put them on slides, and then we can perform a bunch of different experiments looking at how microinfarcts actually change things in the brain at a more molecular level. For example, down here, what I'm showing you is there's a microinfarct located here, and all this red is actually blood leaking out of the vasculature. And blood's very toxic to the brain when it gets out of the blood vessels. And we can also look at, for example, dying brain cells near the microocclusion. So again, you can see the microsphere located here, and all of these cells are brain cells that are dying due to that microocclusion. And then using this model, we can look at different types of treatments as well to look at if we can see if we can mitigate some of the damage of these microinfarcts. So lastly, I just want to thank my lab as well as my supervisor, Dr. Greg Selassie, and the funding agencies that fund the majority of this work. Thank you.
Thank you for the wonderful talk there. I love all the images that you have. So our next speaker is going to be Thomas Edwards, he's a PhD student in human kinetics in his 4th year, supervised by Dr. Lara Kaludi. The topic is exercise for people with multiple sclerosis who use wheelchairs.
All right, are the slides up for everyone? Can someone confirm that before I begin?
Confirm.
Perfect. So thank you for that introduction. So I'm going to shift gears a little bit and kind of talk about some of the more clinical research that we do in our research lab. So in the past, it's been shown that exercise training for people with multiple sclerosis can be very beneficial for improving symptoms, or excuse me, reducing symptoms and improving health outcomes. So we've shown that exercise can improve both cardiovascular fitness as well as muscular fitness. It can also be used to improve walking ability as well as improving balance. And as I mentioned, can also be used to reduce symptoms common to MS, such as fatigue and depression. And while all these benefits are very beneficial for those with multiple sclerosis, it's important to acknowledge that a lot of this research has taken place in those with more mild disability. And as a result, non-ambulatory people with MS or those who use wheelchairs are often omitted from these exercise training studies. So it's estimated that around 25% of people with MS will use a wheelchair at some point during their disease. And current exercise programs do not account for the unique needs of these individuals. And as a result, wheelchair users are often not engaging in regular exercise. So we have this problem where those who potentially need exercise, effective exercise programs the most are receiving the least amount of research attention. So my research goal is to actually improve exercise programs for people with MS who use wheelchairs. And one way I hope to achieve this goal is to investigate adaptive exercise equipment for these individuals. So if you imagine a wheelchair user, they would need to use adaptive equipment due to their physical limitations. So if you imagine something like a treadmill or a stationary cycle that you might find in your typical gym, Given the physical limitations of these individuals, those types of equipment may not be appropriate, and we need to be able to investigate machines, exercise equipment rather, that can be used regardless of disability. However, there has been no investigation on the appropriateness of adapted exercise equipment, and as a result, we conducted what we call the RAMP study, where we tried to figure out which piece of exercise equipment would be appropriate for those with MS who use wheelchairs. So I have pictured here 3 pieces of adapted exercise equipment. Now, I don't have enough time to really describe what each of these pieces of equipment are, but the really take-home message I want to emphasize with this equipment is that all 3 of these pieces of equipment can be used from a seated position, or ideally reducing risks of injury during exercise. So what we did with this RAMP study that I mentioned was we used this machine in the middle here to really gather the physiological response to exercise. So what was happening when an individual was exercising on these different machines, and we compared the 3 machines to each other to really identify which might be most beneficial for improving health outcomes. However, we also want to include a participant's perspective, because that's also something that's very important when you're designing an intervention. It's not just enough to look at the physiology of the machine. You need to look at how an individual responds from a symptomatic point of view, as well as how much they enjoy the piece of equipment if you're trying to make a long-term intervention. So in summary, what we did with the adapted exercise sessions was we looked at the physiological outcomes, we looked at the patient's perspective, as well as the symptomatic outcomes, and we kind of synthesized them together to hopefully identify appropriate exercise equipment for this population. With this data, we're able to make exercise recommendations, and then ultimately, with this data, we hope to improve exercise programs for people with MS who use wheelchairs, which, as I mentioned, is my overall research goal. So again, I'd like to thank the committee for inviting me and sharing my research, and thank you for your time.
Okay, great. Thank you very much. The next speaker is the NICE Lab's very own Attika Pirwani. She is currently, she finished her BA in psychology in 2020. She's supervised by Dr. Nafisa Ismail. And the title of the talk is The Impact of GI Symptoms on Gray and White Matter in Adolescence.
Thank you for the introduction, Kevin. Can everyone just see my slides?
Yep.
Perfect. So today I'm going to be discussing my work on GI symptoms and the effect they have on gray matter and white matter in adolescents. So to start off with, let's discuss the gut-brain axis, which is formed by the central nervous system and the enteric nervous system. And they work together to allow for bidirectional communication between the two in order to control for physiological functions. In addition to this, the gut-brain axis has been found crucial for the maintenance of cognitive performance. Therefore, whenever there's a disruption between this axis, you're going to get problems within the brain and the gut. And one of these disturbances is the presence of gastrointestinal symptoms, which can include but are not limited to things like nausea, bowel inconsistencies, and pain. And these symptoms are commonly found in disorders such as IBS and IBD and happen to impact more than 20 million Canadians. In addition to this, these disorders are most prevalent in children between the ages of 4 to 18. So right there in that middle of that age range is puberty, which is a critical period of development where significant brain reorganization and remodeling is occurring, which also plays a role in cognitive development. So the importance of this pubertal period became apparent in our current study as we saw various structural changes in adolescents who are experiencing these GI symptoms. So what we found within the gray matter volume is that participants who experienced these GI symptoms had actually a decreased gray matter volume in regions that are part of the limbic system. But we also saw that there was an increase in gray matter volume in these same participants in regions that are associated with the so-called pain matrix. And the role of this is for pain processing. We saw an additional decrease in white matter volume in areas of the brain that are usually activated during cognitive tasks involving working memory, language, time perception, executive functioning, and emotional processing. So taken together, this study is a further representation of understanding the neurobiological changes that are associated with GI symptoms, which can also be predicting factors in cognitive impairment. So what we found was that these GI symptoms were in fact influencing gray matter and white matter among regions that are involved in cognitive function and emotional modulation, which may have enduring effects in later life, such as the development of dementia. I'd just like to take a moment to thank my supervisor and everyone else that worked alongside with me on this project. Thank you.
Okay, great. The next speaker here is Daniel Nicholas L. Kodzi. He's a PhD graduate of 2020. He's in Dr. Michael G. Schlaumacher's lab. And the title is A Redox Chemistry-Based Function for Parkinson's Disease-Linked Parkin Confers Direct Antioxidant Activities in Mammalian Brain.
Okay, hi everyone. First of all, I'd like to thank the committee for organizing this event today and allowing us to present our work. So for the past 6+ years of my training in the Schlaumacher lab and extending post-graduation, my research has focused on familial and genetic cases of Parkinson's disease, specifically due to the mutations in the Parkin gene and the Parkin protein, which was first discovered in 1998. Patients with early onset Parkinson's disease due to Parkin mutations are a little bit different than sporadic cases of PD, whereby they develop the disease with an earlier age of onset, which is before the age of 40, and the loss of dopamine cells in their substantia nigra results in the cardinal motor symptoms, which is typical to all cases. However, they do not develop cognitive decline as the disease progresses. So basically, the questions that we wanted to tackle was, what is the essential function for Parkin in human brain, where its loss results in the progressive degenerations of dopaminergic cells? To tackle the question, we investigated Parkin's function and behavior in over 60+ human brains. And what we observed, which you can visualize in panel A, is that as early as mid-age, the Parkin protein is completely absent in the soluble fraction, where proteins are usually active and exert their function, and has completely shifted into an insoluble fraction, where proteins are usually found aggregated and have fallen out of solution. And through logistic regression modeling, which you can see in panel B, we've established a strong correlation between age in human brain and a loss of Parkin solubility and aggregation. Additionally, in parallel, again through logistic regression modeling, we've established a strong correlation between age and an increase of toxin levels in human brain. So basically, based on these observations, we've hypothesized and established a new function for Parkin as an antioxidant, where it's able to reduce the toxin levels in human brains, which results in its oxidation and aggregation. And doing so, it's able to maintain a homeostasis in cells, specifically in dopaminergic cells and neurons, and avoid cell death. These observations were further reinforced from what we've seen and studied specifically in early onset PD human brains due to Parkin deficiency. And in these brains, because of the loss of Parkin, we've seen a significant increase in toxin levels compared to age-matched healthy controls. Additionally, when we compared Parkin antioxidant capacity to glutathione, which is a known and the most abundant antioxidants in cells and tissues, Parkin was much more efficient at reducing toxin levels when we compared to glutathione, which was no different than control. So in our research, what we're able to do is establish a novel function of Parkin as an antioxidant, which was ignored by the field, where in young human brains, Parkin is soluble and is able to exert its function. And during the normal aging of human brains, we see an accumulation of toxins where Parkin exerts its neuroprotection effect to be able to reduce the levels of toxic buildup and in turn becomes oxidized and insoluble. This key finding is crucial to the development of robust PD animal models which are currently lacking in the field and which we have done and characterized in our lab, and I'm not showing them for the sake of time today. And additionally, we're able to test cause-directed therapies, namely through brain parkin delivery, whereby parkin is able to exert its antioxidant function in the dopamine cells and is able to halt or slow down the progressive loss of these cells, specifically for early onset PD patients. With that said, I'd like to thank Dr. Schlossmarker and all the Schlossmarker Lab members, as well as our funding agencies, namely the Parkinson's Research Consortium and the PD community and patients for their continuous support and inspiration. Thank you.
Okay, thank you. So our next speaker is Catherine Healy, a second-year Master of Neuroscience at Carleton University, supervised by Dr. Andre Anne Ledoux and Dr. Matthew Holohan. The title of the presentation is Investigating Brain Network Connectivity and Health-Related Quality of Life Following a Pediatric Concussion.
Hi, everyone. So I'll start out. I'm currently a second-year Master's of Neuroscience program in my second year of a Master's of Neuroscience program at Carleton. I'm also currently a research assistant on the PCARE plus MRI study. This is a study through CHIO, and it's part of an ongoing, was an ongoing RCT that was looking at if early resumption of physical activity after a concussion would associate with reduced risk of ongoing symptoms. So what we did was we investigated whether this physical activity actually changed neurophysiological outcomes at 4 weeks using MRI measures. So we completed MRIs at 72 hours and at 4 weeks following a concussion. And in total, we did an MRI on 75 concussion participants and 30 orthopedic injury or control participants. And that number is what completed all of the scans at 4 weeks. So I'll give you just a brief overview of what a concussion is. So it's characterized as an injury to brain tissue that can be caused by a jolt, a blow, or an impact that causes the brain to move rapidly within the skull. This can cause neurochemical as well as cerebral blood flow changes in the brain. And these microscopic changes to structure and function are what have been associated with the different symptoms of concussion, ranging from emotional to mental to sleep to physical. The three brain networks that I specifically look at are the default mode network, the salience network, and the central executive network. The default mode network is turned on when you're not focused on an external task. The executive network is turned on when you are focused on an external task, and the salience network seems to direct the behavior between the two. In theory, these two networks should be anti-correlated, the default mode network and the central executive network. But after concussion, it's been shown that their activity seems to coincide with each other, which is demonstrating some dysfunction following a concussion and may be associated with some of the symptoms that you see following a concussion. Specifically, one of the symptoms that I'm looking at is health-related quality of life. So that's a measure of a child's perceived well-being in relation to health. So at 4 weeks following their concussion, along with the MRI, they completed a PQL measure, which is a measure of health-related quality of life. And suffering a concussion has been shown to negatively affect health-related quality of life for months after the injury, even after no symptoms are there. So it's a very good marker of potential brain dysfunction that's still occurring even after the symptoms have resolved themselves. So specifically for my thesis, I'll be investigating whether measures of health-related quality of life are associated with network connectivity dysregulations at 4 weeks following a concussion. What this research could potentially do is identify a link between the measurements of quality of life and network connectivity, which could lead to potential cost-effective indicators of connectivity dysregulation following concussion. So what that could mean is a child could attend the emergency department, do the health-related quality of life scan. We could say, okay, maybe there's a dysfunction within the default mode network and find targeted treatments based off of that measure. Okay, thank you very much.
Okay, thank you. So our next speaker will be Min Zhang. He's a PhD in neuroscience student in his 5th year, supervised by Dr. Paul R. Albert. Title of the talk is Treating Post-Stroke Depression via Optogenetic Activation of the Serotonin System in Mice Models.
Thank you very much. Just let me get this going. Great. All right. Thank you for the introduction. Today, I will briefly talk to you all about post-stroke depression and what our lab is working on to improve its treatment. Stroke is the leading cause of disability in Canada. In 2012, over 300,000 Canadians were living with the effects of stroke. Post-stroke depression is the most common mental disability following stroke and affects 1 in 3 stroke survivors. Post-stroke depression slows post-stroke recovery, increases mortality rates, and greatly decreases the quality of life for patients. The most common treatment is a class of antidepressant medication known as SSRIs. SSRIs are frequently prescribed due to having comparatively milder side effects to other antidepressants, but they still exist. Their mechanism action is comparatively complex and requires around 3 weeks between starting treatment and achieving remission in only 50% of patients. The question I want to ask in my research is, can we cut out the middleman and skip all of this? By going directly to the serotonin, can we remove this lag time and potentially these side effects? Serotonin is a neurotransmitter found throughout the brain that is significantly decreased in depressed individuals. Almost all serotonin in the human brain comes from the dorsal raphe nucleus, this small region in the midbrain. Luckily for us, this system in mice is very similar, and decreasing serotonin also results in depression-like behavior from them, and they also have a dorsal nucleus. This makes them ideal for testing treatments before approving them for human trials. In our lab, we use a method called optogenetics to activate the serotonin system. It is a relatively new technique that has revolutionized the field of neuroscience. Through gene editing, we can program the dorsal raphe of mice to turn on in response to light. We bred mice with a serotonin system that responds to blue light. We then produced post-stroke depression-like behavior in our mice without harming their mobility. We then used a micrometer thin optic fiber to deliver this light to the dorsal raphe nucleus to activate the serotonin system. We have found that even brief these mice then undergo a series of basic tests designed to determine their levels of anxiety and depression with either a normal or activated serotonin system. We have found that even brief minutes-long activation of the serotonin system was enough to produce antidepressant-like behavior in our mice, and that through repeated light treatment over days, a more persistent antidepressant effect emerges that may suggest they are recovering from post-stroke depression with this treatment. For us, the next steps are to determine just how permanent this recovery is and exactly which parts of the brain we need to activate to produce this result. Now, while we have no plans to ever stick an optic fiber inside a human, our research can help determine the precise locations to stimulate in less invasive clinical treatments, such as transcranial magnetic stimulation, for example. The more specific we are in our treatment, the less side effects there will be. And we are working to one day have a non-invasive treatment to post-stroke depression that works within seconds and with minimal side effects. Thank you for listening.
Okay, thank you very much. Our next speaker is Mikhail Ladouceur. He's a first-year master's student in neuroscience program, supervised by Dr. Diane Lagace. The title of the talk is Stimulation for Stroke Recovery.
Can everybody see my presentation?
I have you in app mode at the moment.
Okay, perfect. Thank you so much.
Yep, full screen's good.
Perfect. So as previously mentioned, my name is Mikhail, and my work really focuses on the stimulation for stroke recovery. So simply said, strokes are defined as the interruption of blood flow, and this results in severe damage to the brain. And this is extremely important in today's society, as thousands and thousands of North Americans not only succumb to stroke, but are now also living with a long-term disability associated with the illness. So fortunately enough, there are several studies that are now looking into means and therapeutics to improve recovery. And of importance, ongoing clinical trials are now using brain stimulation and have in some cases shown to be beneficial in improving recovery. So these studies really rely on the use of this paddle, which emits transcranial magnetic stimulation to the brain. And this really results in the activation of several cell types, which is suggested to increase recovery. However, there are still a lot of limitations to such studies, and the scientific community doesn't necessarily know which cells are being activated and how these cells can contribute to recovery. And unfortunately, these are questions that are extremely difficult to address in patients. However, there are some preclinical methods that have been developed to determine the role and function of cells. So specifically, there's optogenetics, as previously mentioned. So this is a light-sensitive approach, which really relies on the overall expression of proteins depicted here in green on specific cells of interest, and then shining light on these cells through an optic cable to illuminate the cells and determine their function. And this is a method that I've used for my own study to determine the role, as well as whether the stimulation of astrocyte, which is a cell population that's quite frequently found surrounding the stroke, was sufficient to improve recovery. So in addition to optogenetics, we also looked at mice behavior, and we did so through a task called the cylinder behavioral task. And this is really a task where we put mice in this cylindrical cylinder. It's a glass cylinder, and we can really look at how the mice will actually use their paws. So this first video that I'm showing you is one of a mouse that received the stroke. And I think that we can clearly see that it preferentially uses one paw over the other. So in this case, using its left over its right. However, when we stimulate the mice, so when we stimulate the astrocytes, we see something that's quite interesting and quite different. We actually see at this point that the mouse uses both of its paw in a more or less equal manner. And overall, this seems to suggest that activating astrocytes is in fact beneficial for post-stroke recovery. So finally, I would just like to acknowledge some of the organization, such as the Canadian Partnership for Stroke Recovery, as well as the Brain and Mind Research Institute, which allows students like me to conduct research with mice in an attempt to find new therapeutics for patients living with stroke disabilities. Thank you so much.
Okay, excellent. So we have our last but not least, Haley Geertsma, a third-year PhD in neuroscience student, supervised by Dr. Max Rousseau. The title of the talk is Studying the Effects of Nuclear Alpha-Synuclein in a Novel Mouse Model.
Thank you for the introduction. So I will jump right into this. So Parkinson's disease is a neurodegenerative disorder primarily characterized clinically by motor and non-motor symptoms. This is including resting tremor, slow movement, rigid muscles, among many other clinical symptoms. It's also characterized by the death of dopaminergic neurons, primarily in the brain regions called the substantia nigra and the striatum. Another hallmark of Parkinson's disease are Lewy bodies, which are protein aggregates found in the cytoplasm of neurons, and these aggregates are primarily composed of the protein alpha-synuclein. While these aggregates are hallmarks of Parkinson's disease, recent research has shown that actually the nuclear accumulation of alpha-synuclein is associated with disease and might be a driving mechanism of Parkinson's disease. And so our lab wanted to focus not on these cytoplasmic aggregates, but more so on the increased nuclear localization of alpha-synuclein. So what we did is we generated a new mouse model that has an NLS, or a nuclear localization signal tag, on the alpha-synuclein of the mice. And so with alpha-synuclein, it's primarily located at the presynaptic space here in red. And so to study its nuclear localization, we added this tag that will pull alpha-synuclein into the nucleus to mimic its increased nuclear localization that we see in disease.
So with these mice, we wanted to do extensive characterization to see what will happen when we increase nuclear alpha-synuclein in mice, and then explore the molecular mechanisms of how this is toxic. So one of the tests that we did in these mice is called rotarod. And so this is a test where mice are placed on a rod that rotates at an increasing velocity. And when the mice fall off earlier, we say that they have more motor deficits. In the mice with increased nuclear alpha-synuclein in the purple and green bars here, we see that they fall off the rotarod earlier than the wild type mice here in black, which suggests that increasing nuclear localization of alpha-synuclein causes motor decline in the mice. We also saw an increased lethality in the mice. So the cohort here in purple with most alpha-synuclein in the nucleus had a 25% reduction in survival by about 1.5 years. And we saw a similar reduction in survival when half of the alpha-synuclein is in the nucleus. And so with this, we see significant motor deficits and a reduced survival in the mice due to the increased nuclear localization of alpha-synuclein. And so further studies will be exploring what's happening to alpha-synuclein in the nucleus and why is it causing these phenotypes. And this will hopefully be used to develop therapies targeting nuclear alpha-synuclein in patients with Parkinson's disease. And so I'd like to thank the Russo Lab, my TAC members, the funding agencies listed below, as well as all the organizers of this event.
Okay, so that was our final speaker, and a big thank you to everyone who contributed. It was great to see you conveying so much information in such a short period of time. I'm now going to hand over the hosting job here to my colleague, Julie.
Thank you, Kevin. Hello, everyone. My name is Julie Wallet, and I'm a second-year PhD student in the neuroscience program in Dr. Batsisaka's lab. My research consists of investigating brain metabolism in autism spectrum disorders. As we have seen many informative talks from this evening, I would now like to take a few moments to read questions and to allow our trainees to speak more on their research, their programs, and their experiences. Before we start, I would like to invite the committee members to turn on their cameras for this session. The first question for today's panel is, what is graduate school like? And you can raise your hand when you're ready. All right, go ahead, Terry.
I can start with a bit of an input. So it's kind of difficult to explain exactly what graduate school is like, but in a way it's very different from the undergraduate experience where you're really focused on one thing with a whole group of people. In graduate school, you really get to choose what you are most interested in and really pursue your own ideas, where somebody working right next beside you might be working on something very different that's very specific to them. So that's been one of my favorite things about grad school.
All right, thank you, Terry. So our next question for our committee is, what is academic research like? So go ahead, Leah. Yes, I can go. Well, I've only done academic research. I don't know what is research outside of academia, but at least the research that I've been doing, I think it's quite challenging. I thought at first intellectually, but it's not just intellectually challenging. It's mentally challenging, emotionally challenging, because it's really doing something that has never been done before. So although you can get a lot of help from your supervisors and your colleagues, it remains something that has never been done before. So it's a lot of trying things and really kind of walking in the dark and trying to find new things, but it's also extremely fulfilling when you find something and then you have the thrill and then you're like, wow, this is great. This can really help people and this can really change something in the world. Great, thank you so much, Leah. It's true, graduate school is certainly a big challenge, but it's rewarding at the end. So our next question for this segment is, how did you choose what lab to apply to? So Go ahead, Kevin.
Okay. So my process for finding a supervisor was a little bit extra from what I've been told, but I ended up finding the emails of supervisors I was interested in all over the country and compiling this big list and trying to get Skype or phone call interviews with each of them. And after I found enough supervisors that I was very interested in their work with, I had a little chat with them and asked some questions that I considered were important to me in my future graduate school experience. And I ended up coming up with this little chart to quantify the experience here. I ended up settling with Dr. Nafisa Ismail because she just seemed like such a joyful person, and the lab seemed great with the funding that we had, and the research questions were very interesting as well.
Thank you, Kevin. And the next question is specifically for Haley. And the question is, how did receiving a fellowship help you with other opportunities? Go ahead, Haley.
Thanks. So I received the Bonnie and Don Poole Parkinson's Research Fellowship at the beginning of my master's. So I had only been a master's student for a few months. And I'd say there were two main things that really impacted me upon receiving this fellowship. So the first was it boosted my confidence. So as a new master's student, it's really difficult. You're kind of thrown into the research environment. And so receiving that award boosted my confidence to further pursue academia. And I ended up fast tracking into the PhD program. So now I'm my third year PhD. The other thing is that it really boosted my CV. And so it was able to make me a more competitive candidate for other funding further along in my degree. And so I was able to receive provincial funding via the Ontario Graduate Scholarship, as well as international funding to travel to learn new techniques.
Right, thank you, Haley, and congratulations on your funding. So the next question is again open to the entire committee. And so the next question is, why was completing a research program in Ottawa the best fit for you? Go ahead, Terry.
Yeah, so I think one of the really good things about Ottawa is there's lots of collaboration around. I really like how, especially with medicine here, we have the Brain and Mind Institute, we have the Stroke Institute, the Center for Neuromuscular Disease, the Ottawa Hospital Research Institute, and you're surrounded by lots of extremely brilliant and driven people. So it's very motivating to work alongside them, and you also get lots of collaboration in between with other grad students. So I really like the environment of Ottawa.
Great, thank you. And kind of as a follow-up to that question would be, what did you take under consideration when choosing your thesis supervisor? I can go ahead. Go ahead. So yeah, for me, when I tried to choose a supervisor, I looked a lot on Google to try to find the best, like how to find the best supervisor, and I really found the proper information. But there's two things that are important: the project that you're gonna like, if you're gonna like it, and obviously your supervisor. And supervisor is really important because you're gonna relate to that person a lot and ask a lot of questions to that person, and they're really gonna direct you and help you with your interests. And so I think it's really helpful when you get someone you have the same kind of vision as them, and you have the same a really good interpersonal relationship with them, and they can critique you in a way that you can improve your work. So I think these are all good things to look out for. Great, thank you, Leah, for your input. So we have one more question for today's segment. This is a two-part question. The first part is, what is the impact of funding or this funding on your research? And the second part is, how will it benefit you in the future? It's kind of a challenging question, but Kevin, Haley, do you guys want to try answer that question?
I can try to answer it. So as students, when we receive funding, it helps give us the academic freedom to guide the path of our project, which is something that we don't normally get that much of an opportunity to do. We have some influence with our supervisors to discuss where our project should go. But when we receive external funding, it really helps us get the chance to really make use of this freedom. And then I think how it benefits you in the future is really multifactorial. So like I said, the confidence boost of receiving an award and having your research acknowledged by someone who isn't you or your supervisor is really rewarding. And then I also think it helps just in the practice of going through the process of writing a grant, receiving a grant, and then really deciding how to best use that funding.
Thank you, Haley. Michael, do you have anything to add maybe to that question?
Yeah, I mean, I think that was a really good answer. I mean, I think the confidence boost in receiving, you know, acknowledgement for your work and for your research is important. And yeah, also, I mean, also the timeline of, you know, a master's and certainly a PhD is sometimes uncertain. So to have sort of funding guaranteed, it can be nice for that, right? You can explore different possibilities in your research. You're not sort of constrained to graduate at a particular time. So yeah.
Great, thank you. So I want to thank the committee for answering all of these questions. As this concludes our Q&A session, I would like to hand over the screen to the uOttawa Brain and Mind Research Institute Director, Dr. Ruth Slack. Thank you.
So thanks very much, Julie. And I'd like to thank all of our co-hosts tonight for keeping the whole program going at a great pace. And also I'd like to thank all the speakers for sharing your exciting data tonight. And also I'd like to thank our partners, so the hospital-affiliated research institutes and multiple faculties from across the entire uOttawa. Thank you for your collaboration and participation. And finally, I'd like to thank the University of Ottawa Brain and Mind team, including Natasha Hollywood, our program manager, Victoria Rache, our communications coordinator, and Candice Fortier, our administration coordinator. And finally, I'd like to thank you for being with us tonight. And as you can see, our trainees are really the driving force behind our research programs, and they're the spirit of our program. So if you would like to support our trainees at the University of Ottawa Brain and Mind Research Institute, please go to the link on our website. The power for change is yours, and click on the link. And if you'd like more information on the Brain and Mind or attend future events, please go to our website or follow us on Facebook or on Twitter. So again, thank you so much for being with us tonight.